Good morning and welcome back to the channel. It's always beautiful to have you here. Today we're going to start our discourse by talking about biochemical identification of microorganisms. Now, there are many ways we use to identify microorganisms, but one of the age-long methods is the use of biochemical methods. And when we talk about biochemical methods, we're asking what exactly is the use of biochemical methods. Now, we realize that microorganisms there are diverse and many different types of microorganisms found in different environments. Now, it's important to note that these microorganisms have what we call metabolic pathways. They have many pathways that they use to survive, and different and diverse different types of pathways. So it's important to note that in order to be able to identify these organisms, viewing them on the microscope is not going to be enough. When you're viewing them on the microscope, you're able to tell their shape, you're able to tell if they have a color, you're also able to tell perhaps if they are gram negative or gram positive, but that's not enough to use to identify them. Now you need to go further to do what we call the biochemical identification. And in the biochemical identification, what you are using is your knowledge of the different metabolic pathways that microorganisms use to survive. So we use that knowledge to be able to identify one organism different from another. Now you find out that, for an example, many different types of organisms possess different kinds of enzymes, and they make use of these enzymes in different types of metabolic pathways to use to survive. And so knowledge of this can help us to use to identify organisms. Some organisms are able to ferment and make use of different sugars. Some can make use of sucrose, some can make use of glucose, some can make use of fructose. Now, knowledge of this can help us to be able to identify one organism different from another organism. So it's these different kinds of knowledge that we put to use while doing our biochemical identification of organisms. Also, there's the issue of hydrolysis. Many organisms are able to break down complex molecules in different ways. Some are able to break down starch, some are able to break down cellulose, some are able to break down lecithin. And these are all complex molecules. So it's important that we use this knowledge, knowing that different microorganisms have different capacities, have different capabilities for breaking down of different complex substances. And we can use this knowledge to be able to identify one microorganism different from another. Also, we can look at the issue of respiration and the use of nitrogen. Many different organisms respire in different ways, meaning that some are able to make use of oxygen, so we know that they are aerobic. Some make use of just minute amount of oxygen, so we say they are microaerophilic. Some also make use of do not even want the presence of oxygen. That is, once oxygen comes into the system, is going to destroy them. So we can look at those organisms are called anaerobic organisms. So our knowledge of the way microorganisms make use of oxygen or do not make use of oxygen can also help us to be able to identify one organism different from the other. And this is important because we realize that there are millions of organisms, and they all have different pathways, and it can really become challenging identifying one organism from another. And so this biochemical method is important. There are also other miscellaneous methods where we can use indole test, we can use citrate utilization test, we can use methyl red test, different tests like that we use to identify one microorganism from another. And this is important. Now, we're going to run a series very quickly within the next couple of weeks where we'll be looking at the different tests that make up about what we call a biochemical battery of tests. Many different tests that we do. So we are going to look at all the different tests one by one. So for today, in the next 1 minute or so, we'll look at Catalase test. Now, Catalase test is one of the tests that we use in biochemical identification of microorganisms, and it tests the ability of microorganisms to be able to break down hydrogen peroxide, which is H2O2, into water and oxygen. Now, this is important, and microorganisms are only able to do that if they possess a particular enzyme that we call catalase. So that's why we call it catalase test. So the presence of this enzyme catalase within a microorganism gives that microorganism the ability to be able to break down hydrogen peroxide into water and oxygen. So it's a simple test. Usually you can run the test on a glass slide, put hydrogen peroxide on the glass slide, just a little quantity, and then maybe 1 ml, 2 ml, 3 ml, just a little quantity, and then inoculate, put your microorganism onto that glass slide. And it's a simple test. If there's effervescence, that's you see gas leaving, you obviously see bubbles, So effervescence, you know that a gas is leaving that reaction. So you can see it evident in bubbles. So once you see that, you know that that organism is catalase positive. And if you don't see such a reaction, you know that such an organism is catalase negative. That's just a very simple chemical reaction, a simple test to use to identify organisms one from the other. And this is very important. It's used to identify staphylococcus, which are catalase positive, different from streptococcus, which are catalase negative. And there are many other organisms like that. A lot of bacillus are catalase positive, and you can use them to differentiate from other types of organisms. So when we are doing our identification of microorganisms, we know that we take it step by step and stage by stage. When we see the organism, we grow the organism on a media. We are hopeful that we're able to cultivate the organism on a media. And when we've cultivated it, we want to do a gram reaction test. When we do the gram reaction test, and we have been able to get what the gram reaction is, whether it's gram positive or gram negative, we also now want to mount it on the microscope and then also look at the shape. Is it cocci? Is it oval? Is it rod shape? Is it spirilla? Is it spirochetes? We look at all these shapes, and when we get these shapes, then we go to the biochemical test. We're going to test one by one the ability of the organism, the presence of particular enzymes or absence of those enzymes in these organisms. And at the end of the day, we get a range of results, and this range of results are what we're going to use to be able to identify the organism. I hope you understood, and we'll go through a series where we talk about the different tests, sugar fermentation tests, Indo test, cytochrome oxidase test, and many, many different tests, biochemical tests that are used to identify organisms. Thank you for listening to this. And remember to like, share, subscribe to our content, and do have a beautiful day.